Hello, my dear students. Welcome to my today's online class on one more topic in paper 7, that is developmental biology and organic evolution. In this class we shall discuss about first part of the phenomenon fertilization. Then in later videos we shall discuss the other aspects. Okay, then so by now you know who I am. You can see here a ovum ready to get fertilized by the sperm, right? A sperm approaching the ovum. Then the second diagram shows the penetration of sperm into the ovum, which we are going to discuss it today. Then the third diagram is about polyspermy, which we will be discussing in our future videos. Alright. Now we shall start our class with a brief introduction being, see, living organisms continue their perpetuation of the race only by the process of reproduction. So their continuance on the earth is possible only through reproduction, which can be either through asexual means or through sexual means. Then there is a great difference between sexual mode of reproduction and asexual mode of reproduction. Okay. Comparatively, the sexual mode is more complex than the asexual mode. Then what is the crucial difference or basic difference between the two is the fertilization. Okay, difference in fertilization. It is the main stage of sexual reproduction which is absent in asexual mode. Now, with this background, let us learn the process of fertilization in detail. Now, what is the definition of fertilization to begin with? The union of haploid male gamete, that is the sperm, haploid in the sense n number of chromosomes, and haploid female gamete, that is the egg or the ovum, during sexual reproduction, and finally to result in a diploid zygote that with 2n number of chromosomes is called fertilization. So the union of haploid male gamete, that is sperm, with the haploid female gamete during sexual reproduction to form a diploid zygote is called fertilization. Now, the fusion of gametes may take place either inside the body of the female or outside the body of the female based on the place where the fusion of the gametes take place. Fertilization is classified into two types. If it is inside the body of maternal parent or the female, it is called internal fertilization, and if it is going to be outside, then it is called external fertilization. Now let us learn about the two types of fertilizations. Now, in sexual reproduction, the male inserts the sperms into the female reproductive tract to fuse with the egg. And if this fusion takes place inside the body of the female, then it is called internal fertilization. Okay. Then, so fusion takes place within the female parent. It is called internal fertilization, as seen in humans and most of the animals like cats, dogs, pigs, lions, etc. And in this type, a zygote, the resultant of fertilization act, is formed within the body of the mother. Okay. So this type of fertilization is called internal fertilization. Then, on contrary to this, there is one more type called external fertilization. When the fusion of the sperm and egg takes place outside the female parent, it is called external fertilization, and only a minority of organisms or animals exhibit this type of gamete fusion like fish, frog, etc. What exactly happens here is the female parent deposits her eggs in a place, and later the male parent ejects his sperms over them in the same place, and outside the body the fusion of the sperm and egg takes place. That type of fertilization is called external fertilization. Then, so the gametes that fuse externally have to face many challenges as far as their survival is concerned, since eggs and sperms are deposited in the external environment. Sometimes it can be hostile too. The chances of fusion are quite less when compared to that of internal fertilization. And one more aspect is the predators may eat the eggs as such, or they may eat of zygote also. So to compensate for this loss, that is mortality either at egg stage or at zygote stage, such organisms like fish, frogs, etc. have to lay hundreds of eggs at a time to increase the percentage of survival of the young ones. Okay. So this is about internal and external fertilization or types of fertilization. Apart from that, you have self-fertilization and cross-fertilization that we shall not discuss here. It's enough if we discuss internal fertilization and external fertilization. Now we shall understand the prerequisite of fertilization. If at all, if fertilization has to take place, what all is needed? What you need is sperm from the male parent or the male of the species, and ovum from the female of the species. So the ensurement of fertilization demands maturity of sperm and ovum, that is gametes, and point number 2, coming together of these cells. The first one, that is maturity of sperm and ovum, okay, is fulfilled during the process of gametogenesis, that is process of formation of gametes in the sex organs of male and female, that is testis and ovary respectively. Whereas the second one, coming together of these gametes, is facilitated by the act of sexual reproduction. Now, before getting into the details, we should understand the state of gametes prior to the fertilization. Now let us see how the ovum is and how the sperm is prior to fertilization. So after maturity, the egg is fully prepared with organized membranes, cytoplasmic particles, and reserve food materials. And physiologically, the egg lies with suspended metabolic state. Please note, prior to fertilization, it is in the state of suspended metabolic state and with reduced glycogen contents. Then, the revival of metabolic activity occurs only after fertilization, which must happen within a specific period of time. See, the viability of ovum is fixed for a particular duration, and if at all if development has to take place, fertilization has to take place within that duration. Okay. So, within the specific period of time, the ovum needs to be fertilized by the sperm. And if fertilization doesn't occur, then within that specific period, okay, the egg undergoes degeneration. The egg undergoes degeneration. There is no way that that particular egg can be fertilized by the sperm anytime. Okay, and for the fertilization to happen, the individuals have to wait for next release of ovum. Okay, then what about sperm? Let us see. Sperm cells are adapted for locomotion. They are metabolically active and obtain their energy from both exogenous and endogenous pathways. Then it has been found out that there occurs synthesis of some proteins within the sperm prior to the act of fertilization. Now, what are the major events of fertilization? It is quite a complex process. So, for the sake of easy understanding, the fertilization process or the mechanism can be understood in three different headings. One is penetration, that is penetration of sperm into the ovum, which we are going to study right now. And then activation, second one, and third one, fusion of gametic nuclei. So, we are studying the mechanism of fertilization under these three headings. In this video, I will be highlighting the various physical and chemical processes concerned with penetration of sperm into the ovum. Okay. Now let us study the first one, penetration. During this event, the sperm and ovum come in contact, which involves a number of physical and chemical processes. Too many things to remember, dear students. Don't get confused. Please listen carefully, and the study materials along with the question bank will be posted to you after this video lecture. So, any doubt you can go through the study material also. So, it is quite complicated. Therefore, with lot of concentration, listen to the lecture. Now, first we shall begin with the physical processes that will be taking place in the first part of fertilization, that is penetration of sperm into the egg. Okay. Now, in sea urchin, which is an echinoderm, okay, penetration is possible due to the interaction of chemical substances called fertilizin released by the egg and antifertilizin released by the sperm. And the reaction between fertilizin and antifertilizin is called fertilizin-antifertilizin reaction, which is similar to the antigen-antibody reaction. Okay. Now, so you can see in this diagram a sperm with the antifertilizin molecules, okay, on its surface of the plasmalemma, and you can see some molecules, fertilizin molecules, where you can find the complementarity in the shapes of the receptor part of fertilizin and the antifertilizin present on the sperm. So this is a picture depicting fertilizin-antifertilizin reaction in the sea urchin. This diagram is not there in the study material. Please jot down here. Okay. Now, fertilizin is a glycoprotein It is produced by the ovum, as I have already told you, with a molecular weight of 82,000 or more. So it is a high molecular weight protein, glycoprotein. That means protein having a carbohydrate moiety. Then it contains a number of amino acids and a polysaccharide. Okay, polysaccharide means sugar. Then the antifertilization comparatively is a low molecular weight protein made up of a number of acid proteins secreted by which are present on the sperm. Okay, plasma lemma of sperm. The fertilizants and antifertilizants combine in a particular manner so as to establish an initial bond to facilitate penetration of the sperm into the egg. Okay, now so the fertilizant antifertilizant reaction is species specific. Please note, very important, fertilizant and antifertilizant reaction is species specific. That means the fertilizant produced by the egg of a particular species of an animal, female individual of a particular species, is identified by antifertilizant molecule of the same male individual of the same species. That's what I meant by species specific. And the reaction more or less resembles antigen antibody reaction. Okay, then Tyler, he said that sperm head is engulfed by the surface of the egg, and he termed this phenomenon as pinocytosis. This is just for your information. And then Colvin, they made detailed studies of events during penetration in enteroneutes, that is protocardate, Secoglocus, and a polychaete, that is Annelida, hydroids, coelentrates, under electron microscope, and they have come out with few conclusions. Now, you can see in the diagram how exactly the sperm approaches and penetrates into the ovum. Okay, so their observations have been compiled here and presented here for your easy understanding. Let us study point by point. First point you can make out: sperm head comes in contact with the vitelline membrane. Please note, in the egg you find the vitelline membrane. Below that you find the plasma membrane. Okay. Now the sperm head containing the plasma lemma. Below that the acrosome. Okay. Containing the enzymes, hydrolytic enzymes. Okay. So the sperm head comes in contact with the vitelline membrane of the egg. First point to be noted. Then the plasma lemma of the sperm head and the membrane of acrosome vesicle unite. Okay. The plasma lemma of the sperm head and the membrane of the acrosome vesicle unite and split in the middle. Okay. Then the acrosomal granules come in contact with the vitelline membrane and produce lesion to it. Okay. They form a sort of cut. Actually, the acrosomal granules contain hydrolytic enzymes. Okay. And in humans, it contains an enzyme called hyaluronidase, which I will be discussing little later. So these hydrolytic enzymes bring about lesions in the vitelline membrane. Then the acrosomal plasma membrane everts into one. Okay, everts into one in Secoglocus and many in hydroids. It depends on different animals. Okay, so many tube-like projections are formed which approach the plasma membrane of the egg through the gap of vitelline membranes. Okay, so this acrosomal plasma membrane everts into the tube-like projections which approach the plasma membrane of egg through the gap of white line membrane. So the plasma membrane of egg also sends one or several finger-like villi. You can see the diagram D, 5.8D, what exactly I am explaining. Okay. Then same diagram again I have put here to explain the further processes. The opposing tubes from egg and the sperm unite, D, figure D, 5.8D, and the acrosomal plasma membrane of sperm and the egg become continuous, you can see in E. And thus a connection is established between sperm and the egg. Alright. Now, sperm nucleus and centriole pass through these tubes inside the egg cytoplasm. Okay. So below the acrosome you find the nucleus, and in the neck region you find the centriole, and the nucleus and the centriole both get into the cytoplasm of the egg. Cytoplasm of the egg is called ooplasm. Okay. Then later plasma membrane of the egg and acrosomal plasma membrane of sperm become a continuous membrane for the resulting zygote. Alright. Okay. These are the various steps involved in the penetration of sperm into the egg. Then, earlier these processes were studied in sea urchins, but nowadays similar events have also been seen in toad, that is an amphibian, cat, that is a mammal, chick, bird, and also in humans. So, this basic procedure, that is during penetration of sperm into the egg, remains same in most of the vertebrates. Okay. Now you have to note down that these studies have given a new insight as far as the process of fertilization is concerned. Formerly, people used to believe that fertilization means it is just fusion of male and female nuclei, but now it has been understood that it involves not just the fusion of two nuclei, but the fusion of two cells. Okay. So far whatever I have explained in 3 or 4 PPTs, all that is about physical processes taking place during the penetration of sperm into a egg. Okay. ನಿಮ್ಮ ಜ್ಞಾಪಕಕ್ಕೆ, ನಿಮ್ಮ ಗಮನಕ್ಕೆ, we are still in the first step of understanding the mechanism of fertilization, that is penetration of sperm into the egg. And in that we have finished part of it, various physical processes that take place. Now it is the time for us to understand the chemical processes. Therefore I told you, you have to make some efforts to study this. It is little bit complicated, but if you study in detail, not a problem at all. Put some effort and study. Okay. Now let us talk about the chemical processes. So nowadays people say that penetration of sperm into the egg is largely a chemical mechanism. Okay. So once this anti-fertilization reaction was forwarded by scientists like Lilly and many other workers, scientists or developmental biologists started collecting more and more information about the various chemical substances which are involved in the process of penetration. Then Tyler, he regarded that sperm contains an enzymatic substance that is he calls sperm lysin, which causes local dissolution of membrane to make the path clear for the sperm entry into the ooplasm. Okay. Then other workers like Hartmann, then Rothschild, then Runstorm, they collectively called these various chemicals as gamones. You must have heard of hormones. Hormones secreted by endocrine glands. Chemicals secreted by endocrine glands are called gamones, hormones. In the same way, hormone-like substances or chemicals elaborated by gametes are called gametes. And they regarded these gametes as active principles involved in the process of fertilization. And the gametes released from sperm were called androgametes. Andro means male. And the gametes released from the egg are called gynogametes. Gyno means female. Okay. Now we have to talk about these androgametes and gynogametes in detail, which form the active principles during the penetration of sperm into the egg. Okay. Now let us talk about precursors of the active principle. So Brachet, a famous experimental embryologist who is known for his induction experiments in frog embryos. So he says RNA compound of low specific gravity which is present in higher concentration at the outside of the gametes forms the precursors of the active principles. Okay. Now let us talk about these different gametes. Chemical nature of these active principles called gametes. Gynogamete 1. It contains a bluish red pigment called echinochrome A. Okay. It remains in association with proteins and it is responsible for liberating gynogamete 2. So each one you have to practice. Gynogamete 1 with its bluish red pigment and what it does. Then let us talk about gynogamete 2. Okay. Gynogamete 2. This is a non-dialyzable lipase. Okay, it's an enzyme then, containing 5% of nitrogen but no protein. Okay. Then it can be precipitated by ammonium sulfate, and if heated for 2 hours at 95 degrees Celsius, it loses its property. Okay. Please try to understand each gamete. Okay. Nature of it, properties of it, and role of each gamete in penetration or in general. Significance of each gamete in the process of fertilization, very important aspect. Then there are 2 gynogametes and there are 3 androgametes. Okay. Androgamete 1. It's a colorless, non-dialyzable colloid which is resistant to boiling and cannot be precipitated by ammonium sulfate. Okay. And is quickly inactivated by trypsin. Then androgamete 2. This is yellowish in color, soluble in seawater, resistant to boiling. Then androgamete 3. It is dialyzable, trypsin resistant compound, soluble in methanol. Okay. Now, how exactly these active principles, gametes, act? See, the success of fertilization depends on a fair equilibrium of 2 sets of gametes. There should be a paka equilibrium between gynogamete and androgamete. Only then fertilization will be successful. Alright. So then later people found out that in addition to gametes, certain other substances are also necessary for successful penetration to take place. Please note, don't get confused. We are in the first step and in understanding the mechanism of fertilization, penetration. In that we have discussed fertilizing, anti-fertilizing reaction, which happens to be the physical process. Now it is time for us to understand the chemical process. In that we have learnt androgamete 3 types, gynogametes 2 types. Now, apart from these gametes, you need few more. The process needs few more chemicals. Okay. In case of mammals, for example, what is most needed is hyaluronidase enzyme, what I was talking to you, and glutathione. Okay. Hyaluronidase is produced by the plasma membrane of spermatozoa. helps to break the cells of enveloping layers of the egg, and glutathione maintains metabolic stability of sperm cells. So apart from gametes in mammals, two more chemicals are needed: hyaluronidase enzyme and glutathione amino acid. Okay, and what they do? Hyaluronidase helps in break the cells of the enveloping layers, that is corona radiata, etc., present outside the egg, and glutathione maintains metabolic stability of sperm cells. Then, so ultimately, successful fertilization thus depends upon the presence of specific quantity of sperm cells which provide the requisite quantity of these chemicals. All right. Then the sperm cells immediately after liberation release AG1, androgamon 1. The production of it continues till the arrival of sperm to the attraction zone of the egg. So first one is sperm immediately after liberation release androgamon 1, and the production continues till the arrival of sperm to the attraction zone of the egg. Then it is responsible for retarding excess activity of the sperm and thereby results in the conservation of energy. So at the same time, the egg liberates gynecomastia 1. Then this excites the sperm movement and attracts them to the egg by positive chemotaxis. Okay? Positive chemotaxis means attracting the sperm towards the egg. The result is the oriented movement of sperm towards the egg. Okay? Then, so immediately after the touch of the first sperm with the egg produces gynecomastia 2. Then it binds or agglutinates the sperm in contact and at the same time provokes the production of androgamon 2 in other sperms which make them non-agglutinated. Then the gynecomastia 2 also initiates the restoration of metabolic activity in egg itself, which is marked by the production of second podocyte. Then the androgamon 2 produced in sperm helps in agglutination with egg to penetrate by dissolving the mucus jelly. Then in others it neutralizes the gynecomastia 2 and thus negates agglutination. Another gamete from the sperm called androgamon 3 obtained only from the sperm of sea urchin is believed to have the power of liquefying the egg membranes. So till now, students, I have discussed about the first step of understanding the mechanism of fertilization, that is penetration of sperm into the egg. Okay. So the role of gametes. Okay. Now, so let me stop here. So the first step, that is penetration of the sperm into the egg, is over. In my future videos, I'll be discussing the other two steps. Okay. And then finally significance of fertilization. In the third video, I am going to talk about the fast block mechanism and slow block mechanisms and polyspermy. Okay. So this is enough for today's class. Thank you for your patient listening. Till my next class, read this. Stay blessed. Thank you.